let's look at these pictures. These pictures are all symptoms of alkaptonuria. Alkaptonuria is an inherited condition that causes urine to turn black when exposed to air, and it causes a buildup of dark pigment in connective tissues such as cartilage and skin, even in the eyes, and and it can also cause people to develop arthritis, have back pain in their joints, and have heart problems and kidney stones. This is an inherited disease, which means it comes from the genes from the parents of the people that are suffering from this disease. Well, how does that happen? How can you inherit a disease? In 1909, there was a scientist, Archibald Garrod, who was studying alkaptonuria, and he made a great connection between diseases and your genes. And all the symptoms that these folks were experiencing were because of a buildup of alkapton. It's a chemical in their body. Most normal people have the proteins that they need to break this down, and it's not a problem. But these people did not, for some reason, did not have this protein, and they couldn't break it down, and it builds up, and you see these symptoms. So the idea that the DNA and the genes are linked to these proteins was kind of born. So what does that mean for us? What's that relationship between your genes and proteins in your body? I've heard it said a lot of times that DNA is the blueprint for all living organisms. But when I think of a blueprint, I'm just thinking about the structure, what's there. But DNA is more than just the structure. It's an instruction manual for the whole organism. It and the DNA you have in all of your cells, it has the instructions for every function that you need in your body. And the cells have a lot of functions to do in your body to keep you alive and healthy. They've got to break down chemicals, they've got to transport chemicals, they've got to get rid of chemicals. And if they don't, we have serious problems. You have all of these types of inherited diseases. But DNA is like a huge owner's manual. So I was trying to think of something that I had that came with a huge owner's manual. I was thinking about my camera, and it came with a huge instruction manual. And I get so frustrated sometimes because I just want to know one quick thing, how to turn off the flash, and it takes me forever to find the information. DNA is kind of like that. It's like the complete owner's manual. But your cells, they don't need the complete owner's manual every time they need something. They just need to know, what do I need to do to perform my function? I just need the little quick tidbit so that I can move on. So that's the relationship we're really trying to look at. The DNA and your traits. The DNA stores all that information. We want to take just a little bit of that information and copy it. We copy it into RNA, and then that gets translated into proteins because DNA can't make the proteins. The building blocks for the proteins are out there. We just have to figure out how to put them together so we can perform whatever function we need to. So I've put in here as we go along, if I was taking notes on this presentation, what my notes would look like. So this is just, as you come up with the notes, this is just what I would be writing down. So from genes to proteins, that's what we're working on. We're trying to get that information out from the DNA and create a functioning protein for our bodies. Okay? Oops, I went back the wrong way. So let's talk about what is a protein. Okay, a protein is made up of subunits. Like DNA, there are subunits, but instead of being nucleotides, the subunits are amino acids. And when you start putting the amino acids together, you call them polypeptides. And when these long chains are put together, they can fold up on top of each other and it makes a protein. And the folding is really important because the shape is super important. If the shape isn't just right, the protein's not going to work, and then the function is not going to work. So here are my notes again, just kind of jotting down a little bit information about proteins. Okay, so what is important to know at this point? The function of most genes is to code for the production of a protein. So we have tons of genes in our DNA. Most of them are to make a protein to do something in our body. And the order of the bases in the gene, so from the first bit, the A, T, C, G, the order of those helps determine how to put together that protein and to make it in just the right way. All right, we kind of talked a little bit about RNA. RNA is really the in-between DNA and the protein. We have to have something in between. So what is it? RNA is ribonucleic acid. It copies and translates what's in the DNA and the genes so we can make a protein. DNA can't do it, so we have to have something that copies and translates. What should we know about DNA and RNA and how they're different? Well, the first important point to know is that RNA, the sugar is ribose, and in DNA, it's deoxyribose. The DNA is missing an oxygen. The second important point to know is that RNA has 4 bases. It's A, G, C, and U, uracil, instead of thymine. Thymine and uracil are very similar, but that's a difference there. And RNA is single strand and DNA is double strand. So I just put that in there, a little summary if I'm taking notes. Okay, this slide is great. It gives you the big overview of what we're going to try to get into. The steps from DNA to protein are listed right here, and there's 3 of them. The first one, transcription. Where does it happen? The nucleus. The genes, the one gene we're working on, is transcribed, it's copied to a messenger RNA, mRNA. The next step, translation, is out at the ribosome, out in the cytoplasm. The mRNA is translated into a protein with the help of a tRNA. And then the last step is the folding, which happens in the Golgi apparatus. The protein is folded into its final shape so that it can be used. So I wrote those down in a little shortened form here. If you're going to write these down like this, I would suggest you leave a lot of room between transcription, translation, and folding, and then you can put in more notes instead of writing it over and over again. So I'd leave a good bit of space between those. So here's the first step. Let's get into it a little bit more specifically. Transcription, DNA to RNA. We've got to copy that instruction. And the way we do that is by copying it to a single strand messenger RNA. And this process is called transcription. And it's just making a copy of the instructions. What happens is the gene that we need, the double helix is unwound, and we just make a copy, a template strand of the DNA. And as you can see, it's just one copy, one strand, single instead of double. And the only difference here is it's all matching up, it's all looking just right, base pairing rules until you get the N. On the right hand side, there's a purple U. The U is replacing the T, and that's the key with the mRNA. What happens to the mRNA once we have a copy? Well, we've got to take it out into the cytoplasm where the amino acids are. It leaves the nucleus through a pore in the nuclear envelope, and it gets a ribosome basically just captures it and is going to start making a protein. So it's down here at the bottom. It's captured by the ribosome. Okay, so I added in a couple of things there in red. Okay, the next step after we have made our copy is going to be translation, step 2, using mRNA to make a protein. The next step, which occurs at the ribosome, is to make a polypeptide protein using the code found on the mRNA. This is translation. So we're going to translate the genetic information that's in the nucleotide language into the language of the protein, which is a sequence of amino acids. So I'm adding in a little bit about translation. Okay, so what is the genetic code? To translate the genetic information in RNA, the instructions, into a polypeptide that can actually do it, a protein, we need a code. So the basic unit of the genetic code is the codon. The codon is a sequence of 3 consecutive bases in mRNA that code for one amino acid. So if the 3 consecutive bases are GCA, that codes for one amino acid, alanine. So each codon specifies only one amino acid. So we put the different codons together, strings together amino acids, and we can make a protein. So each codon specifies only one amino acid. How many amino acids are there? There's 20. So the reason the code is 3 bases put together is that if it was only one base, well, there's only 4 bases, U, C, A, and G. That would mean that there's only 4 amino acids. Well, that can't be it. If we said the code is 2 of the bases, well, that's 4 times 4, which is 16. That's still not enough. We would have some mistakes in there. We would mistake one amino acid for another. So we have to have more. So we have a 3-letter code. So 4 times 4 times 4 gives you 64. So there's 64 possible combinations. There's only 20 different amino acids. So that means most amino acids can be coded for by more than one codon. So how do you know what is what? Let's look at this little chart. On the left-hand side, it says first base in the codon. Then at the top of the chart, it says second base in the codon. And then on the right-hand side, it says third base in the codon. If we're going to look for U, we start on the first base. We find the purple U. Okay, then we go up to the top of the chart. We find the next U. So we know we're somewhere in that first box. And then where are we on there? Well, we find the third U. That's phenylalanine. Okay, but if we look on here, UUC also codes for phenylalanine. So there's a couple of different ones for the different amino acids. There's also start and stop codons. So we know where to start the message, where to start our instructions from. This is another way to look at the genetic code. For some people might like this better than the square chart. How do you do this one? Well, you start right in the middle. You take the first letter. If it's U, you find the big U, then go up to the next U, and then find the third little U straight up, and you have phenylalanine. It's the same thing. So we work from the inside to the outside. All right. Role of transfer RNA. It transfers amino acids. So like any type of language, we need a translator for most of us. So if I'm going to talk to somebody, maybe a Japanese person, I don't speak Japanese, so I'm going to say what I want to say. I'm going to have a translator. They're going to take it from English to Japanese and relay that information. We have the same thing here. We have the information from the genes, and it needs to be translated into amino acids. So that's where the transfer RNA comes in. It's found in the cytoplasm, and each transfer RNA, tRNA, binds to a specific amino acid and carries it to the ribosome. So in the first little picture, there's a square, blue square, that's the amino acid. It's bound to a tRNA, and it has an anticodon on the top. What's that? Anticodon is a triplet of bases that matches. It's for the base pairing rules, it's the matching set for the one that's on the mRNA. So the anticodon recognizes and binds to its complementary codon on the mRNA at the ribosome using the base pairing rules. And it's got the correct amino acid hanging off the back. So they just start stringing together. An incoming tRNA binds to an entry point, an A site, on the large ribosomal subunit. And the two subunits then join together, holding the mRNA and the tRNA in place. So having a little information about tRNA. So translation, we're going to mRNA to polypeptide, still going with the same theme. So how does this whole thing get started? Once the mRNA is captured by the ribosome, we need a signal to start. We have the start codon, which is AUG, and we find that on the mRNA. Then amino acids are added one by one to the growing chain of amino acids in a 3-step process. The first tRNA transfers its amino acid to the incoming tRNA. Then that first one leaves, and the second tRNA moves on down the ribosome. The ribosomes keep moving down until we get to a stop codon. And then we have a couple codons that code for stop. And this is a good place to stop right now.